All right, the last two steps together are called the electron transport chain and chemiosmosis. And together, the process that they do is called oxidative phosphorylation. And this is where almost all of the ATP that our cells produce occurs. So where this step is happening is through a series of enzymes that are embedded in the inner mitochondrial membrane. Inner mitomembrane. And that's true for both of the electron transport chain and chemiosmosis. Remember, in our subsequent steps, we've already used up all of our glucose. We've already released all the carbon dioxide. So we don't have any carbon-containing reactants at this point. Instead, all of our reactants are going to be our reduced electron carriers, NADH and FADH2. So the electron carriers are going to get used up in this step. Okay? And what happens is we're going to finally accomplish passing the electrons to oxygen. Oxygen's going to become reduced. Oxygen is the final electron acceptor during cellular respiration. Remember, the preceding step, the citric acid cycle, happened in the mitochondrial matrix. And so the products, including all of those reduced electron carriers, are in the mitochondrial matrix. The next step, the electron transport chain, is accomplished by a series of proteins that are embedded in the inner mitochondrial membrane. And what happens is the electrons are going to get passed from the electron carriers to these proteins. And then that protein is going to pass it to the next one. So this protein will become oxidized, this one will become reduced, this one will become oxidized, this one will become reduced, so on and so forth, until they eventually pass the electrons to oxygen and it forms water. Okay. So another reactant that we need in this step is oxygen, because that's where the electrons are going to get passed to. And we're going to make water as a waste product. Now, this is metabolic water, and some desert animals can actually survive on the water that's produced by cellular respiration. So what's happening during the electron transport chain, as each of those electrons get transferred, they're giving up a little bit of energy. Okay? So the electrons get transferred from the electron carrier to one complex, to the next, to the next. And every time the electrons get transferred, they actually use the energy that's released to pump hydrogen ions across the inner mitochondrial membrane. So the proteins of the electron transport chain, when they transfer electrons, the energy that's released is used to pump hydrogen ions from the mitochondrial matrix out to the intermembrane space. Okay? And this sets up a concentration gradient. So the useful energy-containing product from the electron transport chain is the hydrogen ion gradient. Still, we really haven't made any ATP at this point, okay? And our electron carriers all got used up. Our electron carriers go back to NAD plus and FADH, okay? And now they can be reused in other rounds of cellular respiration to become reduced and gain electron carriers from other glucose molecules. So when we go to the next step, chemiosmosis, the energy-containing reactant really is the hydrogen ion gradient. So our mitochondria are going to use the energy in the hydrogen ion gradient to produce ATP. The enzyme complex that does this is called ATP synthase. That's a vocab word that you need to know. The enzyme complex that produces most of the ATP during cellular respiration, during the last step of oxidative phosphorylation, is called ATP synthase. ATP synthase is also embedded in the inner mitochondrial membrane, but ATP synthase is not part of the electron transport chain. It doesn't receive electrons, it doesn't donate electrons, it has nothing to do with electrons. What happens with ATP synthase is it allows the hydrogen ions to diffuse back down their concentration gradient into the mitochondrial matrix. And when this happens, it actually rotates, and that rotational energy is actually used to phosphorylate ADP and form ATP. So this process of using a hydrogen ion gradient to do work is called chemiosmosis. And so ATP synthase accomplishes chemiosmosis to make most of the ATP our cells use. I think that the animations on Mastering Biology that go along with your homework are great. I just want to give you guys another animation that shows the process of the electron transport chain and chemiosmosis.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase.
You guys don't need to know the names of the electron transport chain proteins, but you do need to know the name of ATP synthase
and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Let's make sure we have everything filled in our chart. So both steps of oxidative phosphorylation happen across the inner mitochondrial membrane. The reactants for the electron transport chain are our reduced electron carriers, NADH and FADH2, and we also have to have oxygen present. What happens during the electron transport chain is electrons get transferred from our electron carriers through the proteins of the electron transport chain down to oxygen, forming water. And with each passage of electrons, the energy that's given up is used to pump hydrogen ions, which are positively charged, into the intermembrane space. We don't make any ATP at this point, and our electron carriers are now back to their form where they can receive more electrons, okay? And now all the energy is in the hydrogen ion gradient. So what happens in the last step is that ATP synthase, the enzyme, uses the energy in the hydrogen ion gradient to make lots of ATP. And it does this through the process of oxidative phosphorylation. That's what we just described. using the energy of oxidizing our electron carriers and reducing our oxygen in order to provide the energy to phosphorylate ADP to form ATP. So one thing that sometimes confuses people, and it seems obvious when I say it, but just to point it out, the electrons from our electron carriers are negative. The electrons are going from one protein to the next protein to the next, to oxygen to form water. Our electrons are negative. The hydrogen ions that are being pumped across the intermitochondrial membrane into the intermembrane space are positive. And then the hydrogen ions have a high concentration in the intermembrane space and a lower concentration in the matrix, and so they diffuse down their concentration gradient from the area of high to the area of low. And diffusion down their concentration gradient releases energy that's used by ATP synthase to make lots of ATP. So if we compare the amount of energy that's available in glucose compared to the amount of energy that's available in ATP, it's only about 30% efficient, okay? So if we compare the amount of energy released by burning by burning glucose directly and the amount of energy we would get by hydrolysis of the ATPs produced, we've lost a lot of energy. What happened to all that energy? Well, it was lost as heat. Each step is inefficient. And so, again, organisms contribute to the entropy or disorder of the universe by releasing energy in the form of heat and disorganizing the molecules around them. So, we've gone through all these steps of aerobic cellular respiration, but right at the beginning, we said glycolysis could happen with or without oxygen. And in order for pyruvate oxidation and all of these subsequent steps to occur, oxygen has to be present. What happens when there's no oxygen? So the last thing that we're going to talk about is what happens in the absence of oxygen during fermentation and anaerobic cellular respiration.